lectures dedicated to cell-cell communication and cell specialization. These are some kind of broad concepts that it's important to have kind of a general overview of. Okay, so these are all features of multicellular organisms, right? Multicellular organisms take on all these different shapes and sizes, but we all have these features in common. And so first, cell mass, right? Cell mass determines the size of an organism. It's the combination of cell size and cell number. So some general things that we know about this. Cell size varies dramatically amongst organisms, right? Cell number also varies pretty dramatically. And cell number is always a combination of how much proliferation is occurring versus how much cell death is occurring, because both are happening all the time, right? A lot of our cells have a specific lifespan. So we have adult stem cell populations that exist to help replenish those cells. Like, who knows the average lifespan of a red blood cell? 120 days. I say that because a lot of people, like, that's the one lifespan everyone knows. Yeah, 120 days, right? So we have these stem cells called hematopoietic stem cells that function to continually replenish all of our blood cell lineages. We have a whole lecture dedicated to stem cells towards the end of class. It's a subject near and dear to my heart, and I love that lecture. And so I kind of pepper you guys with information about stem cells throughout the semester. What is it called again? Okay, so it turns out that cell number is typically more important than cell size in determining the size of an organism. Right, so one of the best examples of that are salamanders. Salamanders have these huge, oftentimes pentaploid cells. So typically, the higher the DNA content, the larger the cell is going to be. And so they have these pentaploid, ginormous cells that puts like our average cell size to shame, but yet obviously we're a lot bigger than them. So pentaploid salamanders have huge cells. Humans have smaller cells, but many, many more cells than salamanders. Yes, that is the hawk again, and yes, he has Sharpays on him. I censored it. Oh no, I cut this. It goes all the way, baby. If you want to. Someone the other day, I think it might have been Jack. My husband was like, "Do your students even know who the hawk is?" I think so. And so I actually spent a lot of time doing like Google image searches for like silly image of person, and just nothing compared in my mind to the hawk with Sharpays. So if you guys have recommendations for who the new like ridiculous person to make fun of is in your generation, let me know, because I don't know popular culture anymore. Memes. I don't know. I saw some things they all just felt wrong, and I shouldn't put them on there. This feels right to me. Okay. So this whole idea of cell number being a combination of cell divisions and cell death. So right, in our culture, we view death as like, oh, this bad, scary thing. Cell death, we like. It is a lot of cell death that happens is a program. It's an important part of our physiology. It helps make sure that the cells that are functioning in our body are functioning in tip-top shape, right? They're not old and crusty and giving out, right? The cells understand, like, hey, I've done my job. It's time for me to undergo this programmed process of cellular death, and then adult stem cell populations replenish that cellular population so that we have new, healthy cells. So this equation applies to every organism. It applies to organs within an organism or a tissue, right? And that is just the equation for how many cell numbers any tissue or organism will have. And so the different types of cell death, they're typically broken up into two broad categories. So necrosis and programmed cell death. And this field of cell death has had a lot of changes to what's been the old convention. But the one thing that remains the same is kind of the morphological features of necrosis versus programmed cell death. So necrosis is typically what happens to the cell is they swell and then they rupture, right? So they release all their cellular contents to the surrounding tissue. And what happens then is this massive inflammatory response. And so necrotic tissue tends to look really inflamed. It has really stereotyped features that all come from the cells basically puking their contents and the immune system being like, oh, what's happening? And diverting all these white blood cells to the tissue, which leads to this massive inflammatory response, right? So it's, in colloquial terms, it's messy. Like necrotic death is messy. And so for a long time, they thought that necrosis was always unregulated premature cell death, like the equivalent of the cell being killed rather than the cell going, oh, it's time for me to die, and undergoing this programmed process of fulfilling that. And so different things that would lead to necrosis, right, disease or some acute injury, starvation, toxicity. Excitotoxicity is specific to neurons. So excitotoxicity kind of happens when a neuron releases way too many excitatory neurotransmitters. And you see this happening when, most commonly, when neurons have been suppressed and then all of a sudden are overactivated. And so the neuron on the receiving end of all those excitatory neurotransmitters can die as a result of that. One way that happens is, so like anti-anxiolytics, anti-anxiety drugs like Xanax, for example, if people are on a really regular dose of Xanax, what happens is it's depressing your neurons. So that's why they say if someone's taking like a regular dose of Xanax, it's really important for them to taper off. If they don't taper off, if they go cold turkey, they can absolutely have seizures that are brought on by this excitotoxicity, right? That results from all these neurons being like consistently inhibited, and all of a sudden be like, oh crap, I'm like releasing all these excitatory neurotransmitters. So, right, it's my PSA for the day. If you are taking a regular dose of Xanax, make sure you taper off and see a physician. Okay, sorry. So it also turns out that necrosis can be regulated as well. And so they've given this term of regulated necrosis necroptosis, because one of the kind of typical versions of programmed cell death is referred to as apoptosis, right? So it's kind of the fusion of necrosis and apoptosis. Basically, the cells are still swelling and rupturing, but it's done in response to specific proteins being activated in the cell, indicating that it's kind of a regulated process in the cell. It's not just the cell rupturing, right, due to too much salt in the environment or something like that, right? It's a regulated process that it turns out can happen, for example, if the normal apoptotic pathway isn't triggered. It's almost like a default pathway, right? So the take home is necrosis, you always have this kind of cell swelling and rupture that leads to the cell releasing all of its contents, leads to, it's messy, and so there's this massive inflammatory response. That can be unregulated or regulated, where there's a molecular program within the cell that triggers the cell death event. Does that make sense? I see blue brows.
There's necrosis and programmed cell death, but there's also programmed necrosis.
Regulated. It's a little bit different. So programmed implies that there's this program in place, playing a little bit of a semantics game here. But basically, and the reason I'm saying regulated and not programmed is because programmed cell death implies something different is happening in the cell. But necrosis can be regulated in that there is a molecular program within the cell that triggers the activation of different proteins that fulfill the cell death, which is really different than just having a neuron inundated with excitatory neurotransmitters and that just causing chaos in terms of what ions flow into the cell, right? Okay. Or if you've ever if you're ever working with cell culture, right, and you put way too much salt in the media, what happens is those cells will just first, right? Because they try to equilibrate their salt concentration. And so what ends up happening is excess salts just lead to basically all this water like basically rushing out of the cells, and they die. I may know a lot about that because it might have happened at my very first lab assistant job. I was making something called phosphate buffered saline, which is what, if you're doing an experiment with cells, you wash them with phosphate buffered saline. I was supposed to be making 1x phosphate buffered saline, and I might have had a math error and made 10x phosphate buffered saline, and might have led to a number of postdoctoral scientists' cells all dying, and then not liking me for the remainder of the year that I worked there, which I understand. Salt concentration and buffers. Okay. So, programmed cell death, sometimes abbreviated PCD. The morphological differences in what happens to the cell is really evident if you're looking at cells in a petri dish or in tissue. They have differences in what happens to the structures when necrosis or PCD is happening. When programmed cell death occurs, the cells do something called blebbing, which basically, here's whatever, a normal cell. Blebbing means like it'll start to like have these bulges that come out of it, and they call those blebs. Basically what's happening is you're going to learn all about the cellular cytoskeleton. There's like this scaffold of cytoskeletal proteins on the inside of the cell membrane. And when programmed cell death occurs, it pokes holes in the cytoskeleton, and cytosol, just fluid, just kind of rushes in, and it creates these blebs. That's molecularly what's happening to create those blebs. So these cellular blebs happen. And the cell starts to shrink inside. It's kind of eating itself from the inside out. And the DNA fragments, so if you actually look at the nuclear DNA, it's breaking into pieces. And the other thing that happens is there's all these eat me signals that the cell starts to put on its surface. Those eat me signals are for phagocytic cells of our immune system that will come and basically phagocytose that entire cell. So it eats it before it explodes, essentially. So that's why this is kind of neat and tidy. It has this blebbing, this crazy blebbing event that happens. DNA is fragmenting, so it's not metabolically active anymore. It's not doing any transcription or translation. All cellular activity kind of stops. Just things are getting broken down. And then the immune system comes. Yeah? They don't really have to. I heard. White blood cell. Right? Will come and eat up that cell before it ruptures. Make them a little friendlier. We like our white blood cells. So, right, so what happens then is you don't have this massive inflammatory response. The cells undergo this nice programmed cell death, they're consumed, and none of the other surrounding tissues are affected. Right? When you have inflammatory responses, that can kind of damage the surrounding tissue as well. The different types of programmed cell death, there's apoptosis. You're going to learn a lot about apoptosis in one of the final lectures of this class. We're going to talk about the different molecular mechanisms that govern that process. Anoikis is a specific type of cell death when a cell that lives in an epithelial sheet leaves that epithelial sheet and undergoes programmed cell death. So one of the last slides has these different kind of general types of cells. An epithelial cell is a cell that resides within basically a layer. So it's it's basically connected to its neighboring cells. More drawing, yes. Drawing situation isn't that ideal. Nothing is that ideal on this room. My benching for the day. So the inner lining of our intestine, right? Our intestine has this cool architecture where it forms these crypts. So this would be like our gut lumen. So gut lumen, so this is where food that's being digested passes through. It's also where like your gut microbiome lives. There's a lot of food, bacteria. And the cells that immediately line the lumen are called the gut epithelium. So each of these, right, these are all cells. And there are different types of cells within the gut epithelium. Lecture 5 talks a lot about epithelia and those tissues. But in order to understand what an oikos is, you kind of have to get it now. Okay, so these are all cells in the epithelium, and in the gut has really cool physiology. There are gut stem cells that basically live at the base of the crypt, right? So we'll call this our gut stem cell. The inner lining of your gut is replaced basically weekly. So if these gut stem cells stop dividing, your whole gut lumen would be gone in like 7 to 10 days, right? So it's really proliferative. So what happens is these divide, they create like teenager cells that are called progenitor cells. So they're just finding their way still, figuring out what they're doing. Because there are a bunch of different types of cells within the gut, right? There's absorptive cells, there's secretory cells. And so these create these progenitor cells that move here, and then they undergo the process of cellular specialization. They become differentiated as they move up this epithelial layer. When the cells get to the tip, they're like, okay, our work's done here. They release. They basically release from the layer and undergo the cell death. Undergo oikos. And that's basically to make sure that this process of digestion happens faithfully, and that most of our immune system actually resides in our gut because we have this massive gut microbiome there. So part of the other thing the gut's doing is making sure that the gut microbiome stays in the lumen and doesn't invade into the tissues. So there's a lot of stuff going on in the gut. And so all those processes are really labor intensive, right? And so basically, constantly producing new cells to do those functions means that they're going to be functioning in tip-top shape, right? So you can't just keep producing new cells without having some cells undergo programmed cell death. Otherwise, your gut would like start to bulge out of your gut. You have all these extra stuff, right? Okay. So that's an oikos. An oikos is a specific type of programmed cell death that occurs when a cell within the epithelium leaves its neighbors. Yes?
Is that programmed cell death typically occur at the end of the cell's life cycle?
Oh.
Happening during the division?
So, life cycle, cycle in terms of mitosis?
Just in terms of like for necrosis, does that happen like any time?
Oh, okay. I see what you're saying.
Like, the end with
Yes.
the job?
You got it. This is the equivalent of someone
That's the way.
dying from like whatever natural causes means, like they've lived a long and productive life versus someone like, I don't want to say anything because someone in here will, right, like some traumatic early death, right? Yeah,
So the closest would be like the latter.
the traumatic early death, yes. Yeah, not programmed, it can be regulated, but it's not programmed. It's supposed to happen that way. Okay. Oh wait, there were 2 other things to talk about. Cornification. Has anyone heard the term cornification before? Yeah. What tissues undergo cornification? Yeah, skin, hair, nails. So skin, we'll talk a lot about it in the context of skin. So your skin has a ton of layers in the epidermis, right? Which is just the epithelial external part of the skin, right? There's also a dermal layer. The skin is a really complex tissue with lots of different cell types. But the outer epidermis is just multiple layers of this type of cell called a squamous cell, flat pancake cells. At the bottom layer, the layer that's right above like the dermal layer, and that's where your stem cells reside. So they're going to start producing all the cells that are going to make the upper layers. But as those cells mature, what happens is they are no longer metabolically active. So when people say like, oh, your stem cells are dead cells, they kind of are, right? They're not when I say metabolically active, I mean they're not undergoing transcription, translation, no new proteins are being made, or really active. What's happening is all the cell contents are replaced by a structural protein called keratin, which basically just gives these cells structural integrity. So they're like functional corpses, right? They're not alive, but they have this really important function, structural function. That's cornification. And autophagy or autophagy, tomato, right? Auto, self, phagy, eating, self-eating. So it's basically cells kind of eating themselves from the inside out. We'll talk a bit more about that in a future lecture as well. The Nobel Prize, was it last year or the year before, was actually given to the scientists who elucidated the cellular mechanisms of autophagy. It's becoming kind of, we're understanding more and more about how important autophagy is for a lot of different normal cellular physiology. So this beefy beef cow has a mutation in a gene called myostatin. Myostatin is a gene that its job is, during development, it helps regulate how many muscle precursor cells get to be created, right? So during development, at some point, there are these regulatory events that say, okay, I think we have enough muscle precursor cells, it's time for them to differentiate into muscle now. And myostatin's in charge of that process. So with this mutation, it's a loss of function mutation, which means that myostatin is not active. So you get this massive pool of muscle precursor cells. So it ends up leading toward causing animals to have way higher muscle mass. So this is then basically bred into beef cattle to create beefier cows, right, for the beef industry. This dog called a whippet, which are like little greyhounds. They're racing dogs. So here's a normal whippet. Here's a whippet with one copy of the myostatin mutation, so it's a little bit beefier. And here's the whippet with the homozygous mutation, like They've identified this mutation in humans as well. So it turns out that it's been identified in families of athletes. So if you do a Google search for myostatin mutation humans, you'll see like one of the images of this like little toddler, like beefy toddler with these massive hamstrings. Yeah. Okay. So the next topic we're going to talk about is regulation of extracellular structure. Right, so people always talk about cells, but you can't really talk about cellular physiology at the exclusion of the matrix in which they reside. So the extracellular matrix is extracellular, so it's outside of the cell, and matrix, it's a matrix of these big molecules that form a structure that's really important for the cells in a number of ways. So in this image right here, the kind of flattened pancakey things, oh yeah, my black nail, I'm gonna lose that one. Anyway, sorry, I don't know, I just distracted myself with my own nail. So the flattened pancakey things, those are the cells. It's a type of cell called fibroblasts. You'll learn more about fibroblasts later. They're one of the most common cell types in our connective tissue. And they form this kind of spread out structure. So here are the fibroblasts, like everywhere you see the flat pancake, but everywhere you see these rope-like structures, that's the extracellular matrix. Those are the proteins that are surrounding these cells and do a lot of important roles. And the cells are the ones that actually secrete those extracellular matrix proteins. It's like they build the house that they then live in. All right, and so it keeps the organism intact, right? And it allows for coordinated function. So there's a lot of different functions that the ECM is important for. Here's kind of a laundry list. So mechanical support and defense. There are a lot of organisms that the extracellular matrix is actually the outside of the organism. So like arthropods, their exoskeleton, that's extracellular matrix. So that's an important example of mechanical support and defense. I was actually at a talk at the Institute for Regenerative Cures over at UC Davis this morning, where one of the PIs there was talking about her work on cardiomyocyte progenitor cells, right? So heart muscle progenitor cells. And she's looking at the extracellular matrix in two regions of the heart, the sinoatrial node and the lateral ventricle, and found that they're structurally totally different. And in the sinoatrial node, there's a lot more fibrous proteins that makes it more rigid, which her hypothesis is that's providing more structural support and protection for those cardiomyocyte progenitor cells to make sure that they are able to divide efficiently and produce the cardiomyocytes that are necessary for actually the pacemaking function of hearts. So it's not just on the outside of arthropods, it's all over our body. They have important roles in helping protect and regulate the cells around them. Adhesion for cells and tissues, this is what a lot of cells stick to. So whether like epithelial cells, on the bottom side of any epithelial layer is a specialized extracellular matrix called the basal lamina. And so all these cells, while they stick to each other, they're also stuck to the basal lamina, which equals basically ECM epithelial sheets. So most cells, if not all, have extracellular matrix that help support them and help regulate their growth and function. If you lose the ECM, you're going to lose structural integrity of the cells, and they are not going to function in the same way. Substrate for cell and organismal movement. When cells move, they need to have a substrate to push against, right? To exert force over. It's the same thing when you walk, you exert force over the ground. The ground for cells is going to be the extracellular matrix. They feel their way around that ECM when they move. Regulation of cell growth and function. This happens in a lot of different ways. A lot of times, so we're going to talk towards the end of lecture today about cell-cell communication. And in some types of cell-cell communication, cells release proteins out into the extracellular space, and those proteins act as signaling molecules, have to move through the ECM. Part of the way they move through the ECM is with the help of extracellular matrix proteins that help shuttle the proteins through the ECM to get to their target cells. In some cases, they found extracellular matrix proteins actually binding to receptors on the surface of cells and triggering signal transduction. So the ECM actually deserves its own couple lectures in this class. It doesn't get it, but it deserves it. It's important. So I will give it a few more slides. So vertebrate ECM. In vertebrates, there are two main categories of macromolecules. Fibrous proteins. So fibrous proteins, think rope-like proteins. Think this, rope-like fibrous proteins. So in general, when people are talking about cells or proteins, they'll refer to proteins as typically either being globular, so just like a little discrete glob, like hemoglobin, or fibrous, so kind of more of a long chain fiber-like structure. So fibrous proteins, there are a ton of different proteins in different ECMs. Here's a couple of the most common that you'll hear of: laminin, collagen, nitrogen, there's a lot of different collagens, and fibronectin. And there's also glycosaminoglycan, which is abbreviated as GAG, polysaccharides, and proteoglycans. So these are either big sugars or proteins with sugars connected to them. And they give a different quality to the extracellular matrix. Fibrous proteins confer strength, and glycosaminoglycans, they form what's often referred to as the ground substance, which I've always thought of Jell-O when I think of the ground substance. So they form kind of this gelatinous structure. So typically ECMs have both fibrous proteins and some ground substance. It's just they can have different ratios, right? And if you shift the balance to fibrous proteins, you're going to have like a rigid, more tough structure. If you shift the balance to GAGs and proteoglycans, you're going to have a little bit more wiggly structure. It's my ECM units. Okay, so the one other area where you see another important feature of ECMs is in bone, right? So terminology, sorry. When GAGs are covalently linked to a core protein, that's when they're referred to as proteoglycans, right? So in case you're confused about proteoglycans, in this image here, green is protein and pink is polysaccharide. So yeah, if you have GAGs linked to a core protein, it's a proteoglycan. So another really important molecule to know for vertebrate ECM is calcium. I'm not actually sure if it's preferred to say appetite or appetite. I like appetite because the way you can remember it is bon appetit. Somehow came up with that. I don't remember if it was the Raphael or Jessica, but yeah. So calcium appetite is what provides the hardening agent for the extracellular matrix of bone. So bone, most of the bone that when you look at this, you're looking at extracellular matrix, right? That cells lay down. So there are 3 major cell types of bone, and they all have a role in building and remodeling. That's vertebrate ECM. Arthropod ECM, it also has fibrous proteins that give it its leathery structure. It has one primary GAG that's called chitin. And then instead of having calcium appetite as a hardening agent, it has calcium carbonate. I was just reading about the calcium carbonate. Does anyone know of another structure in the ocean that relies on calcium carbonate as a hardening agent? What's that? Oh, I don't know. Do they? Like I actually know one correct answer, coral reefs, right? And so coral reefs are these really cool organisms that secrete, like the reef that you look at is mostly exoskeleton, right? That's produced by these little coral larvae that attach and then secrete these massive exoskeletons. And they also produce calcium carbonate as a hardening agent, but if the pH drops too low, they can't absorb their own calcium carbonate. So one of the issues with climate change and increasing temperatures, or actually the CO2 that's released in the air, is that I think it's estimated that like 1/3 to 1/2 of the carbon dioxide that has been released either by human activity or fossil fuel burning has been captured by the ocean. And that leads to acidification of the ocean. So it's dropped, I think, over the last 150 years from like almost 8.2 to like 8.07. And that acidity makes it harder and harder for our reefs to retain the calcium carbonate. And the issue there is the coral reefs are one of the most important ecosystems of the ocean, right, and therefore the world. They have a lot of really important roles. Another fun fact, coral reefs actually get their color by another organism called dinoflagellates that are famous for bioluminescing. There's a lot of different types of dinoflagellates though. They integrate in with the coral reefs and produce all these different types of colors. Okay. If you are someone who really likes to see structures, I was always that student. You may not be that student. It's okay. I'm not testing you on structures. But if you like to see structures, this is an example of what a gag is. If you're like, what does glycosaminoglycan mean? Gag is a repeating disaccharide unit, and one of the saccharides is an amino sugar. That's the meaning of glycosaminoglycan, right? And so chitin is the simplest gag, and it's I know in bio 2 you guys are taught about chitin when you learn about fungi ECM. Bio 2, supposed to remember that? I don't test on fungi ECM. Okay. Plant variations in ECM cell wall. So plants, right? Their cells have cell walls that are that reside outside of the plasma membrane. So one big difference with the plant cell wall is it's basically almost all sugar. So it's primarily cellulose and pectin, and encased or like polysaccharides, right? So polysaccharides are just long chains of monosaccharides. They're sugar polymers. Okay. And then final extracellular matrix we're going to talk about are prokaryote variations. So prokaryotes being either bacteria or archaea. Bacteria, if you're talking individual bacteria, they produce a cell wall that's primarily composed of peptidoglycan. It's kind of a specialized polymer made of sugar and amino acids. And archaea have a different version of that that's referred to as pseudopeptidoglycan. So that's an individual bacteria, right? So it produces its own individual extracellular matrix that is a peptidoglycan cell wall. The bacteria also reside within biofilms, right? And so when bacteria form biofilms, which is a colony of bacteria, they secrete kind of a collective ECM. The collective extracellular matrix that's shared by all bacteria, they still have their own individual cell walls. They have their own ECM. But the collective ECM is called EPS, which stands for extracellular polymeric substance. It has all sorts of macromolecules in there, DNA, protein, polysaccharides. The DNA and EPS, right, when we think about DNA, we think about, oh, okay, genes being transcribed and translated. This DNA is not being transcribed or translated as far as we know. It is functioning as in a structural capacity, right? Because DNA is a big old polymer too, and so it's just functioning in a structural capacity to help do the things that ECMs do, right? So they help lend structure to the biofilm. They help support its growth. They help bacteria within the colony communicate to other bacteria in the colony. Right, peptidoglycan, ECM of an individual bacteria, EPS, ECM of biofilms, which are composed of many bacteria. So biofilms is basically extracellular matrix for a population. If you haven't learned about biofilms before, slimy films of bacteria, you can find them all over. I think more and more microbiologists are learning that it's more common for bacteria to live within biofilms than to live as individual discrete units. They can be really beautiful. So here's a biofilm in a stream in Yellowstone. Tooth biofilm, this is how I get my kids to brush their teeth. I'm like, no, there's bacteria having a party on there. We don't do this, kid. And where they're really dangerous is hospital settings. So there are a lot of, there've been a lot of issues where when, for example, someone is getting like an artificial pit, right? Or something artificial put into their body. If it's not completely sterile, has bacteria on it, and it forms a biofilm within an individual, the thing that's tough is that they're really resistant to antibiotics. So antibiotics, they target proliferating bacteria. So if the bacteria, right, like one of the things that's most commonly targeted is construction of the new peptidoglycan cell wall, right? So if a bacteria is dividing and it can't create that cell wall, that new bacteria isn't going to get to survive. So that's how antibiotics a lot of times will work. But if you have a bacteria that's just like, no, I'm just chilling, I'm not dividing, the antibiotics are useless. So that's one way that biofilms make antibiotics ineffective. Another way is by creating this kind of, this colony of bacteria, it makes it hard for the antibiotics to actually penetrate, to get access to the bacteria that are in inner part of the biofilm. So a lot of times what happens is that they'll try really strong IV antibiotics. That doesn't solve anything. So a lot of times what happens is they have to surgically remove whatever it was that they implanted that has the biofilm. Because it's really scary. It can lead to sepsis really quickly. So bacterial infection. So when bacteria are within a biofilm, they become really adherent and they cooperate with one another. Are you guys ready for a disgusting picture? If gross medical pictures freak you out, close your eyes. This is like one of the tamest. So that is a biofilm being picked at on a person's infected foot.
Dang.
Disgusting, and you don't want that to happen to you.
Damn, that's not the best.
Okay, moving on.
That's not what's that.
Hey, if you do a Google image search for biofilm in hospital, you will see why that was like one of the tamest pictures. I'm not going to read through this. This is just if I was creating notes of the last however many slides, this is how I would have summarized it. So there it is for you. You're welcome. Okay, regulation of cell adhesion. So most of the cells in us have to adhere to survive. So one of the things that happens when cells adhere is they receive survival signals, right? That's how our bodies kind of maintain this program of adhesion. It's not just that we need our cells to adhere, we do, they make it so that cells, when they stop adhering, die. And that's important, and that's one of the things that goes awry when cancer metastasizes, right? So if a tumor is malignant, so that means it's cancerous, but hasn't metastasized yet, that means that tumor is still local. It's in one place. And that's important because that means that it can be, in theory, surgically removed. If it's metastasized, that means that cells within that tumor, which is the mass of cells that are adhering to one another, some of those cells have said, I think I'm going to travel throughout the body and take up residence somewhere else. So they release, they de-adhere, right? They become unadhered, and instead of undergoing cell death, which is the normal thing that would happen, they don't. They're able to travel through typically the lymphatic system, which is why if you have a tumor, the first thing that they'll do is check the lymph nodes nearest to where the tumor is, and then going further and further away to see how far the cells might have traveled. Right, so cancer cells ignore a lot of the rules of the body, and this is one of them, which is you're supposed to be adhered, and you should undergo programmed cell death if you do not adhere, right? So adherence is important. Our cells have to stick to something, or else how would we remain like a contained organism? Yeah, okay. Cells adhere to other cells, right? Or like if you're in the epithelial sheet, you're really tightly adhered to other cells. You're also adhering to the extracellular matrix. There are some cells that aren't within an epithelial sheet, so connective tissue cells, like the fibroblast cells that I showed that first black and white image on the extracellular matrix slide. Those cells aren't really connected to one another, but they're definitely adhered to that ECM. And that adherence is important for their function. And here is, right, this is a prettier image of an epithelial sheet. So these gray big cells right here, that's an epithelial layer. yellow stripe below it, that's the basal lamina that they're connected to, right? Special type of ECM associated with epithelium. And then beneath it would be like the fibroblast connective tissue that has a lot of ECM that it's directly adherent to. All right, so when cells lose these appropriate attachments, they're supposed to undergo a breakdown. Any questions? Guys with me? No? You guys want to hear an Ellie knock-knock joke? Have I told you any of my daughter's knock-knock jokes yet? Oh, I should have embedded the video file. It's so funny. So, I think poop is really funny in our household, and so when my daughter talks about poop and make jokes about poop, and same thing with Emmett, I don't tell her not to. So I was with someone recently, and they said something about that. I'm like, was that bad? Like, I think poop is funny, right? Like, what I think about parent here, and maybe, but I don't think so. And so all of her knock-knock jokes incorporate poop, and they're always different, and they're always totally random. So I have this one recorded from the other day. It's like, knock-knock. Banana. Banana, let me in the house. I need to go poo-poo on your hair. And then the next one was like, I don't know, something else. And then it was like, I'm going to go poop on my mom's face. And I was like, okay, well, that's not too far. Because she could come into the room in the middle of the night. You don't know. So that I can't. Okay. And we're back. Regulation of internal aqueous environment. Another feature that all cells have in common. So all multicellular life on Earth maintains an aqueous environment. Like, right? So this is kind of a stripped down version of the lipid bilayer. On the inside of our cells, the cytosol, mostly water. The ECM, yeah, there are proteins and gags, but it's also a lot of water. Do you know what percent water we are as grown-ups?
60.
You guys are good. We are born at 75%, and then we shrivel up and dry out as we live. So the average adult is somewhere between 55 and 60% water. So muscle has more water than fat, it turns out. So people with higher percentage of muscle have more water. And by the time you're old and gray, if you're lucky enough to make it that far, you will be about half water. All right, fun water facts. So most of us have internal controls that regulate both our pH and our ion concentration. Right? This is the general term homeostasis. I think of it as maintaining our internal balance. Our cells are always trying to achieve homeostasis. And luckily, we have a lot of mechanisms to do that without us thinking about it. And it's not the same for all organisms. So I learned the hard way that plants, the pH and ion concentration is determined by their environment. I learned this the hard way when we bought our first house. Like, we signed on the papers the day I interviewed for this job. And I was a fixer-upper. But I was very pregnant with Ellie, and Jack, my husband, is very safety first. He's like, no, you can't breathe fumes. No, you can't do this. It's like, so I couldn't do anything. I mean, I got to hang out with Emmett all the time, which was super fun. But I wanted to help too. And so he's like, okay, the one thing I'll let you do is the plants in the front yard. But you have to wear gloves because, cat poop and toxoplasmosis. Okay. It's good that I have a safety first partner because I am not safety first, and I should be. But it's good for our kids. It's probably part of the reason they've survived this long. So I thought, I'll do succulents because I kill most plants, and succulents I managed to keep alive for longer. We had torn out the plants that were there before. I can't remember the type of, it doesn't matter. And I just, whatever, got some soil and planted the succulents and was super happy. Like, it was a lot of succulents. So it was more than I'd ever spent on plants. So it's the first time. It was amazing. I did. This is beautiful. And then, very proud of myself. And then all of them died. Like, all of them died. I progressed. I was like, what's happening? Like, am I watering them too much? Am I not watering them enough? And then, I asked, I went back to the plant store, the plant store, and I talked to them about it. I said, well, what kind of soil did you use? They're like, it was brownish, black. I was like, okay, so plant soil matters, and what type of plant was there before? And I told him, he's like, oh yeah, they acidify the soil. And so succulents don't like acidic soil, so you just basically acid poisoned all your plants. There's a new succulent garden that's doing okay-ish, because I thought we had to clear out all the soil and put in special succulent soil that is not acidic. So I will never forget that pH of plants is determined by their environment. So, okay, moving on. I know about animal cells, plant cells, I don't know. So some organisms can maintain their own aqueous temperature, others rely on solar energy, right? That's the difference between warm-blooded and cold-blooded. Warm-blooded means your own body can regulate its temperature. Cold-blooded, they're not actually cold-blooded, right? It just means that they can't regulate their body temperature. It's determined by the external environment. Okay, so this is an image of a mouse embryo, which actually looks, if I put up an image of like a cat embryo or a human embryo, you wouldn't be able to tell the difference. Look really similar, early embryonic stages. And so the vascular system has been highlighted in this mouse embryo. And so the point of this slide is just to convey that the water in our body, it's moving and exchanged between all these different water-containing systems of our body. And really the take-home of these two slides, right, is that the Earth is predominantly aqueous, and all multicellular organisms have evolved to have these aqueous chemistries that are regulated in different ways. The end. Now we're going to talk about one of my favorite topics, which is cell-cell communication. So this is sometimes referred to as cell signaling, and this is basically the process of a cell detecting and responding to a signal in its environment. Oftentimes it is a small molecule or protein. And here's kind of the most stripped down version of cell signaling that you'll see. You have a cell that produces the signal, which is referred to sometimes as a signal producing cell. In this stripped down version, we have the signaling molecule. It's like a green starfish. Right, so the signaling molecule is, in one type of signaling, is released into the extracellular space, and then is going to be received by the signal responding cell. And so in order for it to be received, it always has to bind to a specific receptor. So not all cells can respond to all signaling molecules. But if the responding cell has the exactly correct receptor protein to bind to a signaling molecule, the signal is transduced to within the cell. That's why it's sometimes referred to as signal transduction. The signal is the initial signaling molecule, and then it's transduced into the cell, and that can cause a response. In this image, the response is turning it blue. In real life, it can be kind of any cellular process you can imagine is affected by signaling. So it can lead to changes in transcription. It can lead to changes in cell morphology, right? It can affect the cytoskeleton. It can cause a cell to start moving. It can induce metabolic changes. So anything you can think of, this is how it's regulated, is cell-cell communication. And so both single-celled and multicellular organisms use signal transduction. So single-celled organisms, right, they're communicating with other cells in their environment. And bacteria do this all the time, especially in the context of the biofilm. they will use intercellular signals to basically adapt their processes to what's happening in their environment, right? If there's not enough nutrients around, for example, bacteria in a biofilm, a lot of them will become quiescent, right? Which means they'll stop proliferating. Or maybe if they're, if there's too dense of a population, they'll trigger cell death. Multicellular organisms use it a little bit differently. So in us, we have different types of signaling systems. We have signaling systems where cells that are really close to one another signal to each other, but we also have signaling systems that go throughout our body, right? Like our endocrine system. That's where hormones get released into the bloodstream, travel all throughout the body to affect target cells far, near or far. Synaptic signaling, right? My favorite one to talk about. That's signaling within the context of our nervous system. That's my brain telling my hands to do like jazz hands right now, right? The signal is originating in my brain, and just like that, it's out of my hands doing jazz hands. So, right, signaling in multicellular organisms is a way to coordinate the activity of multiple cells in response to the needs of an organism as a whole. It is how our body works. Okay, so we think that cell-cell communication in multicellular organisms evolved from cell-cell communication in single-cell organisms. So for a long time, we didn't understand that prokaryotes even utilize cell-cell communication. That's more of a recent advancement. And so let's talk about what the heck is happening in this issue, or in this image with the green. What are we looking at? So cell-cell signaling in biofilms is referred to as form sensing, right? And the idea is that bacteria, before they become adherent in the context of a biofilm, they're referred to as planktonic. So you have these free-living bacteria. In this experiment, what these people did is what this group did is they examined how much signaling was going on according to the density of the biofilm. And so what they found is that even in a population where you had all these bacteria, you didn't really start to see efficient signaling until they all adhered to one another within the context of a biofilm. And so all these cells are on a slide, and basically the downstream response of the signaling pathway is turning it green. So you see basically no green in 8 hours, a little bit more green, a little bit more green, lots of green. And this is one of the first experiments where they figured out that signaling within the context of a biofilm was totally dependent on the number of bacteria present, which seems pretty intuitive if you think about it. There are really cool examples of this in nature. There's a Hawaiian bobtail squid that has this light-producing organism that it actually isn't producing the light. It's bacteria that form a colony in its light-sensing organ that actually bioluminesce and produce the light. Cool. All right, so take home. Quorum sensing is cell-cell communication in a biofilm. It's responsive to population density. And the presence of this cellular communication in biofilms made the scientific community kind of understand that this idea of sensing in cellular environment and eliciting a cellular communication event based on that existed before multicellular organisms did, and probably the signaling that occurs within us evolved from that more simplistic signaling system in bacteria. So let's take a look at kind of the general mechanics of a signal transduction pathway. And so it's a lot more complicated than this, obviously, but let's ease into it. So this gray right here is the plasma membrane of our target responding cell. right? And instead of being a green starfish, now the signaling ligand is a red circle, right? And so it's binding to a receptor that sits on the surface of a plasma membrane. It's always the first event is extracellular, usually extracellular, there's always exceptions, binds to the receptor, and that interaction between the ligand and the receptor, it causes a change on the intracellular side of that receptor. And that allows it to transduce the message to the intracellular side of the cell. And it leads to this cascade of events, right? So in this version, you have the receptor activating green square. Once green square is activated, it activates green triangle, then green circle, right? So you have these intermediate signaling proteins within the cell. So signaling molecule binds cell surface receptor, signals transmitted through intracellular signaling proteins, and sometimes also these small molecules called second messengers. Like calcium is a common second messenger, and inositol triphosphate, all these small molecules that you'll know and love by the end of class. Should. So ultimately what happens at the end of the pathway is effector proteins are activated. What makes effector proteins different than the intracellular signaling proteins is they are directly causing the effect, right? The cellular response. So maybe it's a metabolic enzyme that's getting activated that actually alters the metabolism of the cell. Maybe it's a transcription factor that then goes in and activates transcription of a bunch of new genes. Maybe it's a cytoskeletal protein that leads to the cell shape being restructured. Okay, so effector proteins have the ultimate effect. And the other thing about the pathway is it has to work together. You lose any one of these pieces and it doesn't function, right? So if we got rid of green square, wouldn't matter if ligand was binding to the receptor, green triangle is not going to get activated. You guys with me so far? All right. So in multicellular organisms, there are 4 general types of signaling. There's something called contact-dependent or juxtacrine signaling. Different name means same thing. Contact-dependent because the ligand is in direct content with the in direct contact with the responding cell. So one example is there are some ligands that are actually tethered to the membrane, so they can't actually be released by the cell. We'll talk about a few different examples in like a minute. Paracrine signaling is a little bit different. In this case, the signaling molecule is actually released by the cell. So it's released, it's not tethered to the membrane, it's released into the extracellular matrix, and it can diffuse through a field of cells to interact with target cells that are a little bit further away. Synaptic signaling is specifically refers to the signaling of neurons, right? So synaptic because neurons release neurotransmitters in this tiny space referred to as a synapse. Their target cell is either usually another neuron, muscle cell, or gland cell. That's synaptic signaling. And then finally, endocrine signaling. In endocrine signaling, the ligand is always referred to as a hormone. So hormones are produced by endocrine cells. They release the hormones that travel through the bloodstream. So this is really different than just traveling through a field of cells. Traveling through a field of cells, that protein or molecule might get 10 to 100 cell diameters away, right? It's usually going to be within the same tissue. Like if a paracrine signaling molecule is released by a cell in the gut, it may be making it over here, but it's not going to a different tissue in the body. If you're talking endocrine signaling, well, it's going into the circulatory system. That's like the highway of the body. It can go wherever, right? So endocrine signaling and synaptic signaling are sometimes referred to as systemic signaling. I always think of it as system-wide. They can traverse multiple systems of the body. Whereas contact-dependent and paracrine, those are local. They're happening in a local part of the body, localized. So, and it all gets back to how far is throughout the body is that, where does the signal originate and where does the response happen? If the response is a neighboring cell or within the same tissue, like 50, 100 cell diameters away, that's local. But if it's synaptic, right, synaptic is, that's traversing the nervous system. That's, right, originating either in the central or peripheral nervous system to target cells that can be all throughout the body, right? I can wiggle my hands, I can shake my toes, I can do, yeah.
So
You want me? Sorry,
for synaptic, so the target cell isn't always another neuron.
No, it can be that, right, that's how I move my muscles, my nerves are terminating at neuromuscular junctions that control, like acetylcholine is released, which we'll learn all about how muscle contraction occurs. But it's nerves that tell, neurons of the peripheral nervous system that tell our muscles that it contracts. And then also that help tell glands to contract as well. Local signaling equals yeah, that's right. So in theory, right, if a gland releases a hormone, it sometimes can affect tissues that are right nearby. Yeah, it just has the capacity. Great question. So now I have images to go over all of these in much greater detail. So contact dependent, again, juxtacrine, the molecule and the ligand that initiates the signaling is in direct contact with the responding cell. So there's 3 flavors. The one that's shown here is the membrane, there's a ligand that's membrane bound. So the ligand itself is tethered to the membrane. It binds to a receptor on a neighboring cell and initiates signal transduction in the cell. Adjacent ECM molecule. Remember when I was talking about ECM, I was saying that there's some evidence that some ECM molecules can bind to receptors on cells and trigger signaling. That would be referred to as contact dependent because the ECM molecule is in direct contact with the cell that it's signaling to. Right, so contact dependent. It's dependent on contact of the ligand. And then there's something referred to as cytoplasmic sharing. When cells are tightly connected to one another, sometimes they have these junctions. If you're animal cells, it's called a gap junction. If you're a plant cell, it's called plasmodesmata. Here's an example of it. So here are 2 neighboring cells. See these little like tunnels in between the cell, and they kind of are like tunnels. So they can share cytoplasmic contents. And these tunnels, it's not like a free-for-all. It's regulated, it's gated. And a big old protein can't make it through, but a tiny, like second messenger like calcium can make it through those channels. And so when I talk more about second messengers, I'm going to revisit this and remind you, like, this is why when you have signaling initiated in this cell, if it leads to a big release of calcium, and that calcium is transducing the signal, the gap junction opens up and that calcium can make it into the neighboring cell. That can absolutely influence signaling in the neighboring cell. Okay. Paracrine signaling, again, the key here is the signaling molecules are released into the extracellular space. And then act locally on nearby cells, typically within the same tissue, right? So these molecules that are being released in blue, they're released from the cell, but they're not entering the bloodstream, right? They're not acting far in line, they're just gonna be affecting cells as far as the protein can diffuse, which is usually a pretty limited amount of space. Synaptic signaling formed by neurons transmit their signals electrically along their axons, right, via action potentials, and ultimately culminates in the release of neurotransmitters on a target cell. So a lot of times what you have happen, right, is central nervous system sends information to the peripheral nervous system, which affects the target cells. And so you have a sequence of neurons and big old axons hitting the ultimate target tissue to get the response. There are some axons that can extend really far, like the giant there are these big axons in giant squids that are like up to 3 feet long. It's totally wild. Yeah, question.
What determines what kind of neurotransmitter is released? What difference are like supposed
Yeah, it's actually a different neuron. So each neuron is specific to a particular neurotransmitter. So you'll have dopaminergic neurons, serotonergic neurons, glutamatergic neurons. So it's really the neuron itself. If that neuron is stimulated, it's going to release whatever neurotransmitter it knows how to make. So, yeah. Getting started off talking about neurons. Someday I'll maybe do some neurophysics class here, but I hear that the instructors are really good. And maybe I'll start a neurodevelopment class. Okay, that would be okay, so last thing we'll talk about for today, right, is just again endocrine signaling. What's happening here is endocrine cells secrete their hormones into the bloodstream for potential distribution throughout the body. And really what determines what cells respond is, again, do they have the appropriate receptor or not? So if they don't have the receptor, they're not going to be influenced by the hormone, right? So those hormones travel far and wide, can influence all sorts of tissues as long as the correct receptors are present. And we will just pick up here next time.